The life cycle of Trypanosoma and its pathogenicity are complex and involve multiple stages and hosts. 1. Life cycle. 1.1. Trypanosoma brucei. This species, causing African trypanosomiasis, sleeping sickness, has a digenetic life cycle requiring both a mammalian host, human or animal, and a tsetse fly vector. The cycle begins when an infected tsetse fly injects metacyclic trypamastigotes into the bloodstream of a mammalian host during a blood meal. 1.2. Bloodstream form. In the mammalian host, these metacyclic trypamastigotes transform into bloodstream trypamastigotes, multiplying and evading the host's immune system through antigenic variation. These bloodstream forms are responsible for the initial symptoms of the disease. When the parasite population reaches a certain density, the bloodstream trypamastigotes differentiate into stumpy forms, which are non-dividing and pre-adapted for transmission. However, recent research suggests that slender forms can also be transmitted. 1.3. Tsetse fly transmission. When a tsetse fly feeds on an infected mammal, it ingests these bloodstream trypamastigotes. In the fly's midgut, the parasites transform into procyclic trypamastigotes, which multiply and migrate to the salivary glands. In the salivary glands, they differentiate into metacyclic trypamastigotes, completing the cycle and becoming infective for another mammalian host. 1.4. Trypanosoma cruzi. This species, causing Chagas disease, American trypanosomiasis, also has a digenetic life cycle, but with a different vector, triatomine bugs, kissing bugs. The cycle begins when an infected bug defecates while feeding, depositing trypamastigotes into the wound. 1.5. Mammalian stages T. cruzi. These trypamastigotes invade host cells, transforming into amastigotes which reproduce asexually within the cells. Trypanosoma cruzi. This species, causing Chagas disease, American trypanosomiasis, also has a digenetic life cycle, but with a different vector, triatomine bugs, kissing bugs. The cycle begins when an infected bug defecates while feeding, depositing trypamastigotes into the wound. 1.5. Mammalian stages T. cruzi. These trypamastigotes invade host cells, transforming into amastigotes which reproduce asexually within the cells. These amastigotes eventually differentiate back into trypamastigotes, which are released into the bloodstream to infect new cells or be ingested by another triatomine bug. In the bug, the parasites transform into epimastigotes in the gut and then into metacyclic trypamastigotes in the rectum, completing the cycle. 2. Pathogenicity. 2.1. African trypanosomiasis. The pathogenicity of T. brucei stems from the bloodstream trypamastigotes' ability to evade the immune system and their direct effects on the host. Antigenic variation allows the parasites to constantly change their surface coat, preventing effective immune recognition. The parasites also directly damage tissues and organs, especially the central nervous system in the later stages of the disease. This leads to the characteristic sleep disturbances, neurological dysfunction, and ultimately death if untreated. The virulence varies between subspecies, with TB rhodesiense generally causing a more acute disease than TB gambiense. 2.2. Chagas disease. T. cruzi infection causes a wide range of symptoms depending on the stage of infection. The acute phase, often asymptomatic, involves parasitemia and can be followed by a chronic phase, characterized by inflammation and tissue damage to the heart, digestive tract, and other organs. The chronic phase can lead to cardiomyopathy, megacolon, and megaesophagus. 3. Conclusion. Trypanosoma species exhibit diverse life cycles and pathogenicity mechanisms, leading to significant human and animal health problems. The ability of these parasites to evade the immune system and cause substantial tissue damage highlights the complexities of these diseases and the need for continued research to develop effective treatments and preventative strategies.